good day. Welcome back to Microbiology, Prescott's Microbiology. So, how are you guys doing? It's been again after some time since I streamed. I think my gaps between streams are, getting pretty big. So, anyway, I hope you guys didn't forget what we last time studied. Last time we were studying the outer structures of bacterial cell wall. Sorry, not bacterial cell wall, the prokaryotic cell walls. And they are tiny structures that are out of these cells, and it also supports it functions, it has functions. So the one that we studied were, pili, fimbria, and the glycocalyx. The slime layers or the outer layers that covers the prokaryotic cells. Specifically, even though we are studying prokaryotes, we usually do focus on bacteria a lot because they have been studied more in detail than the other types, for example, archaeas, etc. So today will be flagella and how it is related to motility. Motility means movement, how do they locomotion and how do they move from one place to another. Of course, you have seen the bacteria that move from one place to another. Not all bacteria can do movement, but of course some bacteria can do and some bacteria cannot. So today we are going to study how the bacteria do movement by flagella. The upper ones that we studied, the pili and the fimbria, they usually are not for the locomotion, usually flagella. And now we are going to study what the flagella is, what are their types, how many are they number, what is their size, and their structure, and their ultra structure means, they are extremely microscopic structure, right to the details. So that, and then we are going to study how the flagella is synthesized, like here. Then we are going to study how the flagella actually do movement, and you are going to be surprised at how much it acts as a propeller, literal propeller that, for example, just think of it that the propellers that are in boats, boats, what do you call the boats other thing. So this is how it looks, I mean the extending part is being cut. This is just the part that is being attached, and yes, the base part is these are the microscopic structures, and this is how it is synthesized, and this is how it does movement. So yeah, this is even more detailed structure of other things than the basic ones that we studied. So this will be covered in today's topic. So don't get bored, it is interesting. You see these tiny bacteria, and let's just study. So before reading it, I will just tell you the general idea of what it has and what does it do. Before that, let me just check if the live is working or not. My voice is okay, everything's okay, perfect. The avatar, I think it shouldn't smile anymore. Let's get serious. Yeah, so flagella is the protruding that comes outside the cell wall. It has a small part that is embedded in the cell wall, and maybe it reaches to the cell membrane as well, but there is a longest part that is out of the cells. Now the flagella is more thicker than the pili and the fimbria, and is longer than that as well, and is high, actually it is lower in number than that of the pili and the glycocalyx that we studied. So see, most motile prokaryotes use flagella for motility. Singular is flagellum, and the length is 15 to 20 micrometer long. Okay, so it isn't that short. The interesting thing about the flagella is that it moves about 100 cells I'm not sure the exact number, but it moves about 100 cells of its size. Like, and we can move. The highest speed of us is moving, I think, one, two, three, six people of our size per minute. No, not six. But there was an exact number of there. Means it is extremely faster than us, even though we are humans and we are complex. Their movement is extremely fast if you compare it with their size. Of course, if you make a starting line for the race and you put a bacteria and us together, of course we are going to cover the longer distance. But if you just compare it with them moving with their size and us moving with our size, they exceed far beyond with in the speed. Comparing, looking at their small size, they move so fast. Especially movement takes energies, and with their small structures, how do they move such long distances that are 100 times or 50 times more than their body length? Now, the bacteria are monotrichous, polar flagellum, amphitrichous, and lophotrichous. These are the types, the classes that the flagella has been divided into, and it's only on the basis of where it is located, where on the bacteria or the prokaryote it is located. So if it is monotrichous, where I think the diagram is below. Now this is just the movement. Wait, I guess this is monotrichous. The bacteria, the flagella can be anywhere on the surface of. Wait, I'll draw it. First, let's just don't show it again. It doesn't click and then cross. Where is my pen? So yeah, trichous they are saying is for hair. Now the monotrichous, consider this a bacteria. Let's change the color to red. Consider this a bacteria. The flagella, if anywhere present, only one in number. It is monotrichous. And polar means it is present at each pole. If you consider this a single pole and this another pole, so the bacteria is supposed to have two flagellum one at each pole. So this is known as polar flagellum. Amphitrichous, wait, sorry. If it is present on both polar and it is amphitrichous. If it is supposed to be only at one pole, then it is polar flagellum because it is only at one pole. I mixed that up. While the lophotrichous has the cluster of flagellum at both ends. Right? It is present in cluster in high number, even if it is present in the polar, and that is why it is lophotrichous. That means polar, amphi, and lophotrichous are kind of the same because it is all related to poles at the end. These are poles. The one is only at one side. The amphitrichous is on both sides. The lophotrichous is clusters on polar ends. It is supposed to be at both of the ends. The peritrichous is simple. It is the cluster over the whole surface. It differs in numbers, how many of them are covering, but it's supposed to cover the whole bacteria, the whole flagellum. The whole bacteria. So these are its general types and general class that you have to remember when you're dealing with the flagella. Now the ultrastructure, I think it will be better if we study it with the diagram. Here. First look at its basal size. It is 22 nanometer, and we have seen nanometer. Okay, it's in nanometers. The length is in micrometer and the diameter is in nanometers. Now this is divided into three parts. The first part is filament. The first part is filament, the second part is hook, and the third part is base. The base has a lot of rings, L ring, P ring, S ring, and M ring. Now the two diagrams that they have shown side by side are in comparison of the gram-positive and gram-negative bacteria. Now I'm sure you guys will be able to guess which one is gram-positive one and which one is gram-negative one. This one, since you can see it has double peptidoglycan layer, sorry, lipid layer, and this is the tiny peptidoglycan layer, and it also has a lot of space. So this is the gram-negative bacteria, gram-negative, while the other one, it has a thickest peptidoglycan layer and it doesn't have additional outer layer at its cell wall, so this is gram-positive bacteria. Now if you look at the gram-positive flagella and the gram-negative flagella, you'll see the complexity of the gram-negative flagella, because it is embedded in multiple layers, while the gram-positive one is just embedded into the peptidoglycan up to the lipid bilayer. It actually goes through to the plasma membrane and not just the cell wall to tightly being embedded in it, since its movement since its function is motility, locomotion. Now the first part is filament. We all know what filament is. Filaments are made up of fibers. The second part is hook. It is the smaller part that is the protruding out of the cell wall, and it only supports the filament and the base. The hook is the part where it supports the filament and base together. The last part is base. Now this one has a lot of rings. This one has only one ring, so it's simple. What is this? Okay. Now you will be thinking the L ring is, as you can see, it is embedded in the outer membrane, the lipid outer membrane in the gram-negative. So it is L ring. The P ring is the middle ring and it is in the peptidoglycan layer, while the S ring is in the weight. I think, yeah. S ring and M ring are both embedded into the plasma membrane. And yeah, of course this is rod, as you can see. Now the functions of all of them are different. For filaments, it extends outside the cell, so it works as the extra surface, while the energy that comes for the filament movement comes from the basal area, and the hook just transfers it from base to the filament. This is just the general structure. And now the mechanism, just think of it as a propeller. Propeller is always moving in the circular direction, like this spring type movement. You will be thinking that the flagella is like this or like straight, so they will be moving like waves, like this and this. But in reality, it doesn't move in waves. It moves in circular rotatory motion, and the speed is extremely high. The base rotates in the plasma membrane. In all of these membranes, the base rotates. And how does it rotate? Even though they have bonding, they don't have that much of a strong bonding, so that it needs a lot of energy for the bonds to break. It just has enough bonding so that it can be intact with the cell membrane, but it can still move. They say the work is just like the fans, this fan that gives air. It has two magnets on both sides, east, west, whatever, two strong magnets, and there is a rotatory portion in between. It doesn't touch the magnet, but it still rotates due to the magnetic field. The same is the working with the flagellar movement. Even though it is embedded in the membranes of the bacterial cell, it can still rotate, and the speed is extremely high. Now if we go below, this is the flagellar reproduction or flagellar repairment or creation. So this will come in the end. Well, here you can see it's always the rotatory motion. Even if you have one flagella, also it moves either in clockwise or either in anti-clockwise. So the movement that it do, the bacteria will move in the opposite direction. So like if it is moving into the clockwise, the bacteria will be moving in anti-clockwise. Means if it is moving to this from this side, the bacteria will move to that side. Like it's the opposite. Flagella is moving in this direction, but bacteria is moving in this direction. So it's that kind of movement. It's never in the straight line. I mean, they know which direction to move on, so they first adjust their facing, the facing direction. So if it is polar flagellum, if it has only one end, so it's going to move forward. So this one, it's moving, wait, it is clock or anti-clock? So it's moving in this direction. So this is anti-clock and this one is clock. So if it is an anti-clock, it's going to do forward run, and if it is an anti-clock, it's going to tumble. Means it's going to move with braking. The other one's also anti-clock, so it's also forward run. Even though they said it's forward run, it's not actually forward. But I think it just means that it will be moving very fast in a straight direction. And now this one has a lot of multiple directions. This one's moving this way, this one's moving this way, and the opposite one are moving this way. So I think this one will be tumbling. Because if you look at it, these two bacteria have the same number of flagella and the same area that where the flagellas are attached, but the movement is different, just because the direction of the flagellar circular movement is different. Okay, I was not talking about the movement. I wanted to show you the energy that comes for the movement of this flagella. Now we have we have different proteins attached to these rings and the plasma membrane. See, now here in this diagram you can see that these are not attached directly. For example, the S ring, now they're at the M ring, right? So this side we have two proteins, motor A, motor B, and this side has flagella and phlegm and 1, 2. They say this is C ring. I mean, this isn't official ring, but this is kind of considered a ring since it's permanently attached to this area and these are not. The only way is, some prokaryotes have ATP as energy source, ATP, and some just use directly the hydrogen plus gradient that is across the cell membrane as energy source. Now it depends on what type of energy type they are using. Usually it's this kind. Yeah. Now the only diagram left is the growth of the flagellar filaments. Now how do these bacteria, the flagellar filaments, grow if it is embedded in the cell wall and it is out of the cell wall? Now things that grow, that are present inside the cells, like organelles or cellular structures, they are present inside the cells, so you will think that they are easy to grow, and you will think that how are the structures that are outside the cell can grow. So the mechanism is simple. The rod that was present in the ring structures of the flagella, this rod, it is actually hollow. It is empty from the inside. And so there is a ribosome. Messenger RNA, of course, comes for the information of the flagella. It has information of the proteins needed to make the flagella. The ribosomes comes and it translates the messenger RNA, and the amino acid chains that are forming, they go directly into the hollow structure of the flagella, the rod structure. As soon as it gets out of the hook area, these areas are inside the cell, so it is made inside the cell just as everything else, but the filament area is outside of the cell. So the making is, the first thing that is present is like if you consider the start of this as, like it will look like something like this. If it is at the start, there will be tiny bit proteins here. Even though this is the end part of the filament, it still starts from here. So the amino acids that will come from inside, from the ribosomes, it will go attached to that, and this area will slowly and slowly go upward, and it ascends. Like it will go up, and the basal area will have more proteins attached to it, and so in this way it will go up, 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 up even more till the length that is needed for it to be reached, because the messenger RNA will not produce, the ribosomes will not produce more proteins for that. Now the flagellin the filament cap protein. Okay, so this area that was already present in here, that was called as filament cap protein. This cap protein is always present, and it is the starting point from where other proteins will be formed. If this filament cap protein is absent, your filament cannot form. And this is just, and this flagellin are the proteins that makes the flagella. So flagellin, the name derived from that. So it attaches one by one to the cap protein, and it length elongates. So this was growth. Now we just have to read it. Okay, so this is the ultrastructure of bacterial flagella. This is the microscopic structure. They are telling us that the arrows indicate hooks and basal bodies. So if these long things are the filaments, so if you look closely, they will look like some kind of hooks. This area, these are embedded in that. And we can clearly see the ends of these flagellas. Ends. Like if you look at here, this clearly shows that this is the filament cap area. Like it is out of the cell. This is the filament head. And if you look at this, it is the end part. You can, I don't know, you call that the starting point or that the... Oh, I didn't know my mic was not miking. Is it fine now? My mic was actually quite away from my mouth. Anyway, I hope it is fine now, because this is how I usually put it. Anyway, so I'm going to read it now, and because also we can see these diagrams. This is 5 micrometer. Micrometer means it is, but the flagellum was longer than these micrometer, where the actual length was written here. Anyway, we'll move 15 to 20 micrometer long. See, it is the, okay, so this is the longer bacteria. Okay, thank goodness it is cool now, because I was starting to get worried. So because the longer flagella is 15 to 20 micrometer, it doesn't mean that the smaller ones will also be 15 to 20 micrometer. There are smaller flagellas. I think the longer the flagella is, the longer will be the movement is. Also, they have shown the number of the pili and these are the pili and the flagella. Flagella are actually high in number. Okay, so I was confusing the numbers before. So this is Pseudomonas bacteria. This is Spirillum, and this one has, as you can see, this is the polar flagellas. Polar. This one, sorry, not polar. What am I saying? Yeah, but polar has only one flagella at the end. So this is Lophotrichus. Not Lophotrichus. Yeah, and this is not the Peritrichus. Now this one is the polar flagellum. This one is the Lophotrichus. And what are these ones? These are the Peritrichus, because the flagella is covering the whole bacteria's body. So you need to be able to differentiate between the flagellas of different bacteria, because it also shows what kind of bacteria this is and what category does it belongs to. Thank you for keep up. So now it's the only part that is left is reading, and we will skip through that. Not, what do you call it? Skip or skim through that. because I also don't like to read. Come on. Come on, why is not moving? Yeah. Man. Okay. Flagella and motility. Most motile prokaryotes move by the use of flagella, singular flagellum, thread-like locomotor appendages extending outwards from the plasma membrane and cell walls. Thread-like means they are very thin. Locomotor means they do locomotion, they help in movement. Appendages are, of course, you can say it's like legs because it helps in movement, and it extends outside from the plasma membrane and the cell wall. Means it is not just the outer structure of the cell wall, it also comes out from the plasma membrane. Bacterial flagella are the best studied and they are focused of this direction. Yeah, that's what I'm saying. Because the bacteria that we usually study stuff of the bacteria, because bacteria are studied highly in comparison to the archaeas, because archaeas are also prokaryotes, but we study bacteria. Now, bacterial flagella are slender, rigid structures about 20 to 20 nanometer across and up to 50 to 20 micrometer long. So this 20 nanometer is the width of it, diameter of it, while this 15 to 20 micrometer is its length, like how long is it in longitudinal. So this is 15 to 20, and I guess this is nanometers. What did I do? Okay. Then the flagella are so thin that they cannot be observed directly with a bright field microscope. So we were talking about capsules, and capsules can be seen without any staining or anything, but the flagella, even though they are higher in number, they are very, like we said, thin and thread-like, so we cannot see it if we don't stain it. Must be stained with special techniques designed to increase their thickness. The detailed structure of flagellum can only be seen. Means even if we stain them, we cannot see them, so we have to make them thick. Bacterial species often differ distinctly in their patterns of flagella distribution, and those patterns are useful in identifying bacteria. Okay, we know that bacterial species is different because their flagellar distribution is different. Like I said, if we know the distribution of the flagella, we can at least put them in different classes or families. Monotrichous means have one flagellum at an end. It is, if monotrichous, if it is anywhere on the bacteria surface, it is monotrichous. If it is located at the end, at one end only, at one pole, so it is polar flagellum. And amphitrichous means you have poles, you have a lot of have single flagellum at each pole. Okay, so that one was lophotrichous. So amphitrichous, now you have poles and you have only single flagellum at both ends. In contrast, lophotrichous have cluster of flagellum at one or both ends. Okay, so it doesn't have to be at both poles. Even if it is there, even if there is a cluster of flagellum at one end, it is lophotrichous. Even if it is on both ends, it is also lophotrichous. Flagella are spread fairly evenly on the whole surface of peritrichous. Yeah, so if it covers the whole body, if it is clusters or if it is in whatever form, it is the peritrichous. Now, flagellar ultrastructures. Ultrastructures mean we studied under the electron microscope, especially transmission electron microscope, because it studies the structure very clearly. Have shown that the bacterial flagellum is composed of three parts. The longest and most obvious portion is flagellar filament, because it is the longest one, which extends from the cell surface to the tip, and then a basal body is embedded in the cell. It is inside the cell, and a short curved segment, the flagellar hook, links the filament to its basal body and as a flexible coupling. I already showed you in the diagram, so we just read it. The filament is hollow, rigid cylinder constructed of subunits of the protein flagellin. Oh yeah, we studied flagellin, the protein that makes up the filament, which ranges in molecular weight around 30,000 to 60,000 daltons, depending on the bacterial species. The filament ends with a capping protein. Yeah, ends with a capping protein, but it is the start of where it will start reproduction, no, growth. Even if it is the end part, it is the start part of the growth. Some bacteria have sheaths surrounding their flagella, some one, okay, not all of it. For example, Vibrio has a membranous structure surrounding the filament. Vibrio cholera has a lipopolysaccharide sheet. So the Vibrio has a membranous structure, while the other one structure composition we know, and that is lipopolysaccharide. The hook and basal body are quite different from the filament. Why? Because it is embedded in the cell, so of course its structure has to be compatible with the cell. The filament can do whatever it wants. Slightly wider than the filament, the hook is made up of different protein subunits. It is made up of different ones, while the filament is made up of only one type of protein, and that is flagellin. The basal body is the most complex part of the flagellum. In E. coli and most gram-negative bacteria, the basal body has 4 rings connected to a central node. The outer L and P rings associate with the lipopolysaccharide and the peptidoglycan layer. That means outer layer to the plasma membrane are L and P rings. The inner M ring connects to the plasma membrane. Now gram-positive has only 2 basal body rings, while the inner ring connected to the plasma membrane and the outer one probably attached to the peptidoglycan. Probably means they're not sure about that. So it's just as we studied. As we've seen in the diagram, so no need to go into detail. Flagellar synthesis. Now, how do you synthesize it? Okay, flagella, even though it is longer in length, it doesn't have that much of a complexity, especially the filament part. So, in that regard, it has only 20 to 30 genes. Okay, so they are saying it's a complex process. To me, including 20 to 30 gene is not a complex part, like it is just a flagella, I mean. But still, I think they consider it 20 to 30 genes are a lot for just a structure formation. Besides the gene for flagellum, 10 or more genes code for the hook. Oh, okay. Okay. So they are saying flagellin, the genes for flagellin aside, 10 or more genes code for hook and the basal body protein. Only 10 or more genes. Other genes are, do we have something here? Okay. Are concerned with the control of flagellar construction or function. Yeah, exactly. Like I said, the structure takes more genes than the functions alone. How the cell regulates or determines the exact location of the flagella is not known. Yeah, exactly. Like I said, the movement of the flagella and the movement of the bacteria is opposite in direction. So they don't know how the cell knows which direction to move or which direction for its bacteria to move. But even though they know, we don't know how they do it. When flagella are removed, the regeneration of the flagellar filament can then be studied. Okay, so it is a reproductive organ. It unless you stuck it out from the bottom, it can regenerate if you remove the filament. Transport of many flagellar components is carried out by the apparatus in the basal body, that is specialized type 3 protein secretion system. We already studied the protein secretion systems. Alright, so this one is type 3. We have studied till type 5. It is thought that the flagellin subunits are transported through the filament's hollow internal core. We talked about the road inside there, right? And it was hollow, so the flagellin subunits move to that. When they reach the tip, the subunits spontaneously aggregate in a direction of special filament cape. Aggregate means they cluster the proteins. Even though the amino acids, when come out from the ribosomes, they are not in aggregate form, they are in chain form. But then here, when it is, a parent of the special of this filament cape, they aggregate, because that's what filament cap tells them to do. I mean, what do you call it in chemistry language? The bonding kind of make them aggregate. Filament synthesis is an excellent example of self-assembly. Yeah, because no one is telling them to move this way, or there is just self-assembly because the bonding makes on its own. Many structures form spontaneously through the association of their component parts without the aid of any special enzymes or other factors. The information required for filament construction is present in the structure of flagellum subunit itself. That means the amino acid chain bond formation is already depending on the flagellum units itself, subunits, right? Because they, just as magnets, it has S side and N side. If you are going to move S in front of the N, of course it's going to get attached. So the same is the case with subunits. They already know what units they are going to get attached to, so it doesn't need enzyme. They just go and attach themselves without the extra expenditure of energy. So this is self-assembly. The mechanism of flagellar movement. Now, how flagella moves? Prokaryotic flagella operates differently from eukaryotic flagella. How? The filament is in the shape of rigid helix and the cell moves with the helix rotation, okay? So the prokaryote one moves with the rotation. Considerable evidence shows that the flagella acts just like the propellers on the boat. Bacterial mutants with a straight flagella or abnormally long hook region cannot swim. Bacterial mutants, if they have straight flagella or if they have long hook regions, they cannot swim, because the only way for the bacteria's flagella to work is in propeller forms, in circular form, in rotation. When the bacteria are tethered to a glass slide using antibodies to fill filament or hook proteins, the cell body rotates rapidly about the stationary flagellum. So they rotate because they don't want to get attached to the antibodies, right? No. We use antibodies and we attach them to filament or hook proteins. And then it is tethered to glass side, means it is attached to that. The cell body rotates rapidly about the stationary flagellum. Oh, because the bodies are... A cell body rotates? Okay, because we made the flagella attached to the glass. The base part of the flagella is attached to the glass. So that's why the only way for the cell to move forward, it rotates, because it is stuck there. So it only is able to rotate. If the flagella was moving in a straight direction, it would have never rotated. It would have been still stuck, just like car. If it is trying to move forward, because it moves in a straight line, even though it is stuck, it won't rotate. But propellers, if you stuck them and they are unable to move, the only way they will move is rotation, because that's the only way they have left, and that's the only type of force that is not anti-forced. If the polystyrene lattice beads are attached to flagella, the beads spin about flagellar axis due to flagellar rotation. Now, if you put beads to the flagella, the beads spin about the flagellar axis due to the flagellar rotation. It spins because the flagella has now something that we can see, and we clearly see how it moves. The flagellar motors, wait, let me show it through the diagram what we are studying now. Consider this a glass slide. Consider these antibodies and this a bacteria. They first said that we put these antibodies here and we tethered it on the glass, means the bacteria is now stuck here. And it has, how is it stuck? Because we put glue-like substance here, and now for glue we used antibodies. Now it is stuck here, so the bacteria wants to move, and the flagella will start to rotate. If it is rotating in that way, the bacteria is going to rotate in this way. So the bacteria is going to change the direction slowly but surely, and the flagella will go to that side and the bacteria will go to this side. This part won't change. This part will still be stuck, but the bacteria will start to move in circles. Now this is one step that we studied. The other that we studied is we took a glass, now we took a bacteria, and we put beads on the flagellar. I think the beads were just used as a markers, because when you stain the flagella, it is so thin and it moves extremely fast, so we can't make the image of the flagellar movement. Even though we are moving, we know it's moving, but we don't know how it is moving because it's so fast. So we put beads because it is heavy and it will have spaces between them, and we know how it is made. So it will move, and it moves in this direction. This is how the after images of the beads will be formed, and so we know that it is propelling. Now, the rotating helical, wait. The flagellar motor can rotate very rapidly. The E. coli motor rotates 720 revolutions per second. 270 revolutions per second. 270 revolutions is not joke. Rotating against your body per second, and 270 at that. In 1 second, how much can you rotate to look at the same glass wall that you were seeing before rotating? It takes us 2 seconds maybe, and these guys, they rotate 270 revolutions per second. So that's how much of a speed does it have. The direction of the flagellar rotation determines the nature of bacterial movement. Monotrichous polar flagella rotates counterwise when viewed from the outside of the cell during normal forward movement, where the cell itself rotates slowly clockwise. See, the polar flagella is moving counterclockwise while the cell is moving counterclockwise. This one, the cell is moving clockwise. The rotating helical flagellar filament thrusts the cell forward in a run. Okay, even though it's rotating, of course the speed is that much high that it is moving forward also, in a run with a flagellum trailing behind. Monotrichous bacteria stop and tumble randomly by reversing the direction. Okay, so this is telling us how the bacteria actually stops. Now, if it is moving in the counterclockwise direction the whole time, and suddenly it moves in clockwise. If it was moving in anti-clockwise and suddenly it moves in clockwise direction, of course the bacteria is suddenly going to stop because it has to change the direction. So it tumbles, it kind of stops. This is their way of stopping from moving. Peritrichously flagellated bacteria operates in somewhat similar way. They move forward, the flagella rotate counterclockwise. As they do so, they bend at their hooks to form a rotating bundle that propels the cell forward. Yeah, they bend at their hooks so that all of the flagella are in one direction. Clockwise rotation of the flagella disrupts the bundle and the cell tumbles. Because bacteria swim through rotation of their rigid flagella, there must be some sort of motor at the base. Now, motor means they must have some energy supply for these flagella to move, and because the flagella is rigid, means it needs even more energy. A rod extends from the hook and extends in the M-ring, where it can rotate freely in the plasma membrane. Because like I said, it's not directly attached to plasma membrane through covalent bond or ionic bond or this kind of bond, not this strong bonding. It is thought that the S-ring is attached to the cell wall in gram-positive bacteria and does not rotate. The PL rings of the gram-negative bacteria would act as bearing for the rotation rod. Means it supports the rod even more, while the gram-positive one S-rings is attached, so it doesn't move, it doesn't rotate that much. There is some evidence that the basal body is passive structure and rotates within a membrane-embedded protein complex, much like a rotor of an electric motor turns in the center of a ring of electromagnets. I already explained that the status, two magnets and one thing is rotating in between because of the opposite forces of the magnets from both sides. The exact mechanism that drives the basal body, the exact mechanism is not known because the structure is so thin and we haven't even found the whole structures. So the mechanism comes after that, so we don't know exact mechanism, but we can guess around that much from what we have seen. The rotor portion of the motor seems to be made primarily of a rod. Rotor means the rotatory portion. The part which rotates is primarily made up of rod. The M-ring and the C-ring join to it on the cytoplasmic side of the basal body, means the lower area, the periplasmic, the cytoplasmic, whatever area. These two rings are made of several proteins. Flagella is particularly important in generating the flagellar rotation. The two most important proteins in the straighter part of the motor are mot A and mot B. So again, these are all the proteins that are helping in rotation. So these form the proton channels through the plasma. Now there are proteins, and now they are helping in the ion gradient transfer. So it helps in energy. Mod B also anchors the mod complex to the cell wall. It also helps in attachment as well. There is some evidence that mod A and Flag directly interact during the flagellar rotation. Means the motor A and flagella means if they two act and forms bonds and whatever, it will help the bacteria to rotate, work. Not directly by ATPase in the case of eukaryotic flagella. Yeah, see, in eukaryotic flagella the energy comes from ATP, but in prokaryotes it is from the gradient movement and the interaction and the tension that is formed because of that. The flagellum is very effective swimming device. From the bacteria's point of view, swimming is quite a task because the surrounding water seems as thick and viscous as molasses. Yeah, bacteria point of view, the water is very thick and viscous. Okay, so swimming is a pretty big deal because if it was that easy, every bacteria would be able to move. Oh my goodness, how much is left? Okay, just a paragraph because I have to, because my charge is ending. And okay, let me see if this is still happening. Yeah. Let's bore to the water through its corkscrew-shaped flagella. Corkscrew. Corkscrew means the basal area of the flagella is like that. And if the flagellar activity ceases, it stops almost immediately. What is this, man? I'm so done with ads. Anyway, they are saying as soon as the flagella movement stops, they stop moving. Despite such environmental resistance to the movement, bacteria can swim from 20 almost 90 micrometers per second. This is equivalent to traveling from 2 to over 100 cells length per second. Means they move about 100 cells their size in just 1 second. In contrast, exceptionally fast 6 feet human might be able to run around 5 body lengths per second. Look at the comparison. Even if we run fast, in 1 second we can only move to only about 5 bodies length per second. But the cells have the ability to move past 100 cells of their size per second. Bacteria can move by mechanism other than flagellar rotation. Okay, so not just flagella is helpful, but other things can also help bacteria to move. Spirochetes are helical bacteria that travels through viscous substances such as mucus and mud by flexing and spinning movement caused by the special axial filament composed of the periplasmic flagella. Periplasmic flagella means the flagella is even inside. Means it is like the flagella stuff, but it is a filament only. It is special type. The swimming motility of the helical bacterium spiroplasma is accompanied by the formation of kinks in the cell body that travels through the length of the bacteria. I think it is talking, the amoeba movement, it kinks, it drops and elongates and kind of that kind of movement because the cell itself is so, what do you call? Kinks is the cuts. So I don't know how it moves by making cuts or forming cuts in the cell body, but it moves by that. A very different type of motility, gliding motility. I think King's one is more strange than the gliding one though, but still is employed by many bacteria. So gliding means they just glide, they move, they slip free, they slip. Cyanobacteria, myxobacteria, cytophags, and some mycoplasma. Although there are no visible external structure associated with gliding ability, it enables movement along solid surfaces at rate 3 micrometers. They are like snakes. I think this gliding motility is like snakes, you won't see any apparent outer structure, how they will move, but they still glide on the and their speed is so fast as well. So I think this is our flagella, and I think we are done with it. So hope you guys have learned a lot of things on the flagella. I thought it was interesting. I thought it was pretty interesting. So hope you guys found it interesting as well. So now let's smile and end it on something funny. But I don't have any funny joke. Okay, first let's just shut up, okay? Don't smile that much. Also, I don't know if my mic was working fine or not. Should I be shocked? Oh. Again, no. Okay, what am I doing? I don't have charge and I should turn this off. The battery? How much was the length of this live? Should I make myself bigger? Ha ha ha. I'm so big now. I'm so big. Wait. Is my conversation even showing? Oh no, it isn't. This is a bad place for me to put it. Because no one can see what was written here. Gosh. I need to have a background for it then, I think so. The invisible thing doesn't work and I have to color it. Anyway, goodbye. Good luck. Today's flagella motility ends.